So haematopoiesis is the developmental process that allows the regeneration of our blood cells every day. This means that every day we need to produce billions of new red blood cells, white blood cells, and platelets, because many of them are very short-lived. For example, neutrophils only stay in circulation for a few hours. So from when we get up in the morning to when we go to sleep in the evening, all our neutrophils are pretty much new again. So yes, it is an amazing process, and even though it leads to the generation of very many cells, it is all reliant on just very few stem cells which reside in the bone marrow, and they just are able to maintain it all. And so one thing that is very interesting to understand is how these stem cells, which are so few and actually also proliferate very little, are able to regenerate all of the blood. Malignant cells are the cells that form the cancer, and the cancers I study in particular is leukemia, which is cancer of the blood. When patients receive chemotherapy, this works really well because it gets rid of the vast majority of the leukemic cells. However, it also selects for very few cells which are able to resist to the chemotherapy. And this is because they're able to metabolize it, or it is because they hide somewhere specific where they cannot be reached by the chemotherapy, or they may be helped by what we call a malignant niche to survive the chemotherapy. And so it is becoming very important nowadays to understand what are the mechanisms that allow chemoresistant cells to survive so that we can target them. So our study revealed that leukemia cells, and particularly T-cell acute leukemia cells, are highly migratory. And they are migratory also after response to chemotherapy. And this is something that we really did not expect, because the hypothesis in the field was that there is some malignant niche somewhere that helps the chemoresistant cells to hide and survive the chemotherapy. And instead what we see is that they're very highly migratory. We're still in the process of understanding what are the mechanisms that regulate this migration, but what we hope is that this ability of the leukemia cells to migrate throughout the bone marrow space is a new target that we can now focus on, and perhaps if we can affect the migration of the cells, we will also make them more susceptible to chemotherapy.